The genetic history of Mexicans offers a profound window into the intricate interplay of indigenous heritage, colonial influences, and modern biomedical implications, reflecting a demographic narrative that is both unique and universally significant. Mexico's population today is a mosaic shaped by thousands of years of indigenous settlement, followed by European colonization in the 16th century, and the forced migration of African slaves during the colonial era. This convergence of ancestries has produced a genetic diversity that is among the most complex in the Americas, characterized by significant variation across regions and ethnic groups. Recent advancements in genomic research have illuminated this complexity, providing detailed insights into the ancestral components of indigenous populations, the admixture patterns in mestizos, and the health outcomes influenced by this genetic legacy. Understanding this history is not only a matter of historical curiosity, but also a critical factor in addressing contemporary medical and social challenges. The indigenous peoples of Mexico, numbering over 60 distinct groups, represent a remarkable reservoir of genetic diversity rooted in pre-Columbian civilizations such as the Aztecs, Mayans, and Zapotecs. Genomic studies have revealed that these populations exhibit levels of genetic differentiation that rival those observed across continents. For example, research published in 2014 examined over 1,000 individuals, including 511 native Mexicans from 20 indigenous groups, and found that the genetic distance between certain groups, such as the Seri in the northwest and the Lacandan in the southeast, is substantial. This differentiation is quantified by an FST value of 0.136, a measure of genetic divergence that exceeds the 0.11 observed between Europeans and Chinese populations in global datasets. Such findings indicate that the genetic variation within Mexico's indigenous groups is comparable to that seen across vast geographic expanses elsewhere in the world. This stratification is further evidenced by principal component analysis, which maps a northwest to southeast gradient in genetic variation, with the third principal component accounting for 0.89% of the variance, nearly 3 times the variation observed along Europe's north-south axis. This indigenous diversity is not merely a statistical curiosity, it reflects millennia of adaptation to Mexico's varied environments. A 2020 study sequencing the exomes of 78 individuals from 5 indigenous groups, Rarámuri, Huichol, Nahua, Triqui, and Maya, estimated that the genetic split between northern and southern populations occurred approximately 700 years ago. This research identified specific genetic adaptations linked to survival in distinct ecological niches. For instance, the Rarámuri, renowned for their long-distance running prowess in the rugged Sierra Madre, carry variants in the BCL2L13 gene, which is associated with enhanced endurance. Similarly, the Triqui, a group from Oaxaca known for their shorter stature, exhibit variants in the KBTBD8 gene, potentially tied to body size adaptations. Other genes, such as SLC24A5, influence skin pigmentation, reflecting evolutionary responses to solar exposure across Mexico's diverse climates. These adaptations underscore the deep historical roots of indigenous populations and their resilience in the face of environmental pressures. The arrival of Europeans in the 16th century, followed by centuries of intermixing, gave rise to the mestizo population, which today constitutes over 90% of Mexicans. This group embodies a blend of indigenous American, European, and to a lesser extent, African ancestries, with proportions varying significantly by region. A comprehensive study of 492 parent-offspring trios from Mexico City reported median admixture proportions of 65% indigenous American, 31% European, and 3% African. In contrast, Mexican-American cohorts in Los Angeles showed a shift to 45% indigenous, 49% European, and 5% African, suggesting migration-related differences. Regional analyses further highlight this variability. In the state of Guerrero, a study found 69% Native American, 27% European, and 1.9% African ancestry, while northern states like Sonora exhibit European ancestry as high as 66.7% to 95%, and central and southeastern regions retain Native American contributions of 37% to 50%. These geographic gradients are not random, but mirror historical patterns of colonization, with European influence stronger in the north and indigenous ancestry more pronounced in the south. The dynamics of this admixture are also shaped by sex-biased gene flow, a legacy of colonial power structures. Paternal lineages, traced through Y chromosome markers, reveal a predominance of European ancestry, averaging 64.9% across mestizo populations, with 30.8% Native American and 4.2% African contributions. Maternal lineages derived from mitochondrial DNA are overwhelmingly Native American, reflecting a pattern where European men intermingled with indigenous women during the colonial period. This asymmetry has left a lasting imprint on the genetic landscape, with ancestry-specific analyses recreating a genetic map of Mexico that aligns with its geography. Northern mestizos carry a higher proportion of European ancestry, while those in the south and southeast retain stronger indigenous signatures, a pattern that genomic tools like ancestry-specific principal component analysis have vividly illustrated. African ancestry, though averaging only 3% to 5% nationally, plays a more pronounced role in specific regions, particularly along Mexico's coasts. Between 200,000 and 500,000 Africans were forcibly brought to Mexico during the colonial era, primarily to work in plantations and mines, leaving a genetic legacy most evident in states like Veracruz, Guerrero, and Oaxaca. In the Costa Chica region of Guerrero, African ancestry can reach up to 20%, a testament to the concentrated settlement of enslaved populations. Genetic studies have identified African haplogroups, such as E1b1a, in these communities, corroborating historical records of gene flow during the conquest and colonial periods. This ancestry has cultural significance, enriching Mexico's multicultural fabric with Afro-Mexican traditions, and biomedical relevance, as it contributes to traits like sickle cell anemia susceptibility, which requires targeted health interventions. The genetic diversity of Mexicans has profound implications for health, influencing traits ranging from lung function to chronic disease risk. Research has demonstrated that subcontinental ancestry affects forced expiratory volume in 1 second, FEV1, a key measure of lung capacity. A shift along Mexico's east-west genetic axis is associated with a 7.3% decrease in FEV1 per standard deviation, equivalent to the lung aging experienced over a decade. For example, an individual from Sonora in the northwest might exhibit lung function markedly different from someone in Yucatan in the southeast, a disparity driven by ancestry rather than environment alone. This finding underscores the necessity of considering genetic background in respiratory health assessments, particularly in a population with such pronounced ancestral variation. Another critical health outcome tied to this diversity is type 2 diabetes, a major public health challenge in Mexico, where prevalence rates are among the highest globally. Genomic studies have pinpointed the SLC16A11 variant, more frequent in individuals with Native American ancestry, as a significant risk factor. This variant, potentially introduced through Neanderthal admixture, highlights how ancient genetic legacies can shape modern disease patterns. The interplay of ancestry and disease extends to pharmacogenomics, where drug efficacy and safety can vary by genetic background, necessitating personalized medical approaches. Beyond disease, adaptive traits like those seen in the Raramuri and Triqui populations illustrate how genetic variation has historically equipped Mexicans to thrive in diverse conditions, from high-altitude endurance to compact stature suited to specific lifestyles. When compared to other Latin American populations, Mexicans exhibit a distinctive genetic profile, characterized by a higher proportion of indigenous ancestry and regionally variable European and African contributions. In contrast, populations in Colombia and Puerto Rico often display greater African ancestry, reflecting different colonial histories, while countries like Argentina show stronger European influence. Mexico's genetic diversity is thus a microcosm of its historical trajectory, with indigenous roots, European overlay and African threads weaving a complex tapestry. This uniqueness invites further exploration, particularly through whole genome sequencing, which could uncover rare variants and epigenetic modifications that deepen our understanding of both demographic history and medical applications. The study of Mexican genetic history is an evolving field with far-reaching implications. It bridges the past and present, revealing how millennia of migration, adaptation, and intermixing have shaped a population that is both diverse and cohesive. For historians, it offers a molecular lens on Mexico's cultural mosaic. For medical professionals, it provides actionable insights into health disparities and treatment strategies. Future research should prioritize comprehensive genomic surveys and community-engaged approaches to ensure that the benefits of this knowledge, whether in healthcare or heritage, reach the populations from which it is derived. Ultimately, Mexico's genetic story is a testament to resilience and complexity, a narrative encoded in the DNA of its people that continues to inform and inspire.